and welcome to IB Biology Notes with Dr. Kimmo. This is A3.2 Classification and Cladistics. This chapter is higher level only, and it's about why and how we're classifying living things into groups or clades. Classification makes it easier to store and keep track of all the information we have about different organisms. You might remember Carl Linnaeus and his system of classification of all living things. That system is still in use, but it's developed quite a bit since his times. Here's an example of how this system can be useful. Now, imagine you've never seen the organism on the right. What can you tell about it? First, it's clearly multicellular. That means it belongs in the domain Eukaryota. It has many features typical of animals, which means it belongs in the kingdom Animalia, not, for example, Plantae or fungi. Upon closer examination, we can conclude that it has a vertebra, so it belongs in the phylum Chordatum. We can also see that it has fur, mammary glands, and some of the properties typical for the class Mammalia. If we follow it for an hour or two, we can see that it's adapted to life in trees, and well, if we dissect it, we can see it has a large brain. This is typical for the order primates. Now, at this point, perhaps we don't know that much about primates, but we know it's a primate. We can read about primates, and we might find out that very large eyes and long fingers are typical for the family Tarsidae. Now we're already at the family level. Now, there's not that many species in this family. The number is very limited, so it's not difficult to finally find out that it's a Carlitosuricta. And once we know the Latin name, we can discuss the species with other people from any language group, and we can, learn more about it. Sometimes classification is not that easy because evolutionary change is gradual. Speciation and the formation of new families, orders, classes, and so on does not happen overnight. Yet taxonomists still want to assign all organisms in taxa. That's the plural for taxon. Sometimes it's difficult to draw the line. How do we determine whether two groups of organisms are similar enough to be placed in the same family or just in the same order? This is called the boundary paradox. Although genetic analysis can reveal probable evolutionary relationships between the species, it's up to taxonomists to draw the line between the levels of classification. Early taxonomy was based on apparent similarities. Similar organisms were placed in the same group. But since Charles Darwin and his theory of evolution, taxonomy has been based on evolutionary relationships. So how organisms are related. There are two important points here. Every organism with a common ancestor is in the same taxonomic group, and all members of a group share the same common ancestor. All members of a group therefore share some traits that they inherited from that ancestor. This allows prediction based on classification. A group of organisms with shared ancestry is called a clade. Clades include all living and ancestral species. For example, on the right you have a cladogram of cetaceans, and the ones marked with an X are extinct. They're still part of the same clade. Clades exist at different levels. Here are some examples. The yellow clade is nested in the blue clade, which in turn is nested in the red clade. So you have three different levels of clades in this illustration. Each species is therefore in several clades. Here are some examples of what a clade is and what a clade is not. Top left, the green box, that's a clade. Same thing with top right, the blue box, that's a clade. Bottom left, the red box, that's not a clade because we're leaving out some groups that should be in that box. Also, bottom right, that's not a clade because we're again leaving out a group that should be in the same box. DNA is essential for unraveling the evolutionary relationships between species because differences in DNA sequences are due to mutations and the mutation rate is roughly constant. Therefore, differences in DNA sequences can be used to estimate when two species diverged. This so-called molecular clock is affected by many things like generation length, population size, and selective pressure, so it sometimes gives us relative rather than absolute dates, but it can be calibrated against fossil data. DNA and amino acid sequences can then be used to construct cladograms. That's branching diagrams that illustrate ancestor-descendant relationships, like the one on the right. A cladogram has terminal branches for individual clades, internal branches for ancestors to two or more clades, and nodes, the point when an ancestral species split to form two or more clades. At the bottom we have the root. Some cladograms are drawn to scale, which means that the distance between nodes represents time. And it's important to note that this cladogram should not be seen as definite evidence, but as the most probable pattern of evolutionary history based on the data that was used. Now, you could use data from just one single gene, but including several genes can make a cladogram more reliable. Modern cladistics can redraw old family trees. Before genetic analysis was available, classification was based on apparent similarities, as mentioned before. Cladistic studies have confirmed much of the traditional classification, but in some cases it has turned out that species in a taxonomic group do not share a common ancestor. Therefore, some taxonomic groups have been rearranged. An example is the foxglove, which used to belong to the figwort family, but has been moved to the plantain family. More than half of the species in the figwort family have been moved to other families after cladistics studies showed that they were actually not as closely related as we used to think. Here's another example of how family trees have been redrawn based on cladistics. Traditional classification into domains was based on cell type, eukaryotes and prokaryotes, two domains. Comparative analysis of rRNA revealed two distinct groups of prokaryotes, eubacteria or bacteria and archaea. Now this cladogram shows how the split between archaea and eukarya occurred after bacteria had split off from the common ancestor of all three domains. This means, for example, that humans are more closely related to archaea than bacteria are to archaea. This three-domain system that is in use today was proposed in the year 1990. That's all for now. See you next time.